All right, just quick programming when we get started. Today's Knowledge Portal and webinar and workshop will feature the Type 1 Diabetes Knowledge Portal with 2 guest speakers, who I'll introduce momentarily. I'll first start with a little background, and then I'm also going to start with some programming notes for folks just joining. Those of you just joining, just so you know, these resources and webinars are held every other month. Our next one's going to be in March, and that one will be really a nice one. We'll have one of the postdocs from Jason Flanagan's lab come talk about a new feature that we'll be having on the portal at the end of this month. So that'll be very exciting. And again, we have them every other month at the same time, 12 noon Eastern time. And if you miss them, we video all these, and they're on our YouTube channel posted to our resource page. So you can always find them there, and it's pictured here if you can't see that. Additionally, on our resource tab, you can also find videos for all the features and functionalities for the portal, updated regularly, along with tutorials and extensive documentation done by our very own Maria Costanzo. So with that, I'm just going to give a little bit of framing and context, and one programming note as we go, because we have 2 speakers today in addition to me. We have a lot to cover, but we're going to try and save time for questions. If you have questions along the way, please pop them in the Q&A box, and we'll make sure we record those and come to those at the end for sure. So with that, I'll just give a little context. Since we're talking about type 1 today, I thought I'd give a little background in terms of how we arrived at type 1, because I think it's a nice context in terms of how all these things come together. Many of you who have attended webinars historically or used the portal before are familiar with our first portal that's almost 5.5 years old now, and this is the Type 2 Diabetes Knowledge Portal. And this resource has over 60,000 visitors, has average of 500 users per week, so it's really well-established activity. And this was really, the cornerstone project from the AMP, Accelerated Medicines Partnership in Type 2 Diabetes partnership that's been going on for the last 5 years, which was a lovely partnership, which we had the pleasure of being involved in, which was between the NIDDK and 5 pharmaceutical partners with a goal to advance the use of human genetics and designing new medicines for Type 2 diabetes. And there are several other AMPs going on in the field as well. We are lucky to be part of Type 2 diabetes. And the mission of this, and I think this was useful because this helps sort of set the stage for how Type 1 diabetes came into the mix, was to generate data for Type 2 diabetes and its complications, make this data accessible, not just to the experts, but to people who can actually make use of it. Create a knowledge portal, the knowledge portal for Type 2 diabetes, that contains a comprehensive phenotype and genotype data for Type 2 diabetes and these complications in a usable and accessible framework. And hopefully this will enable the use of this data to support drug discovery. So that was AMP T2D's mission. And I think what's happened over the course of that is that several other communities and fields have really embraced the idea of making genetic data broadly accessible. And the last year, we've been very lucky to take those portals, some that were for cerebrovascular disease, cardiovascular disease, sleep disorders, in addition to the Type 2 diabetes portal, and combine them into one resource, all powered by the same software and data platform, which we've talked about this webinar, and also add to it Type 1 diabetes. We released this officially at the end of last year, several weeks ago, and is a common metabolic disease knowledge portal. It's a common resource for all of these phenotypes and data for genetic and genomic resources. So today we're going to talk about the Type 1 diabetes resource, which is inside of this resource. And I'll do the tour in both resources so you can see the features and tools therein. And so what the Common Metabolic Disease Knowledge Portal now has in it for you to explore is a rich set of data and traits from over 50 countries and a really multi-ethnic representation of 325 traits from 168 data sets. What's really unique about this that separates it from other large-scale resources is community-driven. It has a lot of unique data and results that you're not going to find anywhere else, and results and methods, run on these data that you're not going to find anywhere else. And that's what we're going to talk a little bit about as we talk about Type 1. This really represents the largest community-based representation of data for common metabolic diseases in an open access framework today. But what's happened in our field over the past 5 years is we've identified a lot of loci, and the Type 2 diabetes and now the Common Metabolic Disease Knowledge Portal makes these accessible to you. But the challenge is we have a lot of loci, but very little in the way of variants, genes, the tissue of action, or the mechanism at these loci. And this case holds true for Type 2 diabetes. We have 402 loci, obesity, coronary artery disease, fat distribution, height, and the list goes on. And you're going to hear about the same challenge for Type 1 diabetes in a minute. So for us, really what we've done with genetic data, we need to move toward function. So over the course of the next year, our goal is to shift and make the resource really a place for the ability to figure out what is the variant, what is the regulatory effect, and what tissue, what is the gene, what is the effector gene at this locus, what is the pathway, and ideally, what is the mechanism so we can ideally make better treatments. And that is really what's happening in the complex trait genetics field in general. And type 1 diabetes is a really strong effort to achieve that goal for it. So what's happening now is that, the AMP type 2D is sort of moved to AMP CMD. And what we hope to do with that effort is to amplify the foundational work of AMP 2D, expand our data and software platform to provide open access to the research community for the data, the methods, the results across all common metabolic diseases, not just type 1 diabetes, not just type 2, but all related common metabolic diseases. Ideally, in addition to that, for this holistic approach across common metabolic diseases, we also want to provide a substrate for community-specific knowledge, because I think that's really what differentiates us. The fact that we work with the communities to represent their results the way they meant them to be represented, but integrated in a fashion that's accessible to someone who's a non-expert. That's really the difference. And ideally, under this model, under this mission, we really want to find effector genes for type 2 diabetes and all related phenotypes. We want to give the best, what is the gene that you want to study in your functional follow-up? If we do this right, we will perform in addition to this, we'll be performing functional characterizations with several of our collaborators, of which you'll hear from 2 today, to determine the function at these gene and loci. And our goal with AMP, the portal, is to make this accessible. So with that, NID's commitment has continued. They funded, as part of AMP, they funded a portion of the first round of AMP T2D. But now for CMD, the commitment continues. They funded the portal for 5 more years of effort to take on the same challenges I just mentioned. They've also funded a host of functional genomics projects for type 2 diabetes. There are 3. They're mentioned here. You'll hear about these over the coming months in our webinars, but this is really to find vector genes for type 2 diabetes and its related traits, and also make these data accessible, generate new experiments, and bring these to the portal in a way that others can use. Moreover, NID's commitment has increased across type 1 diabetes, and which is what brings me to our presentation today, where there are 4 funded entities, 2 of which you're going to hear from today, to generate and represent functional genomics data for type 1 diabetes. And so 2 of these projects, one led by Kyle Gaulton and one by Steve Rich, you're going to hear about in a moment. But this really poises us to not only have a place to make a software platform to make the results accessible to the world, but to work with these collaborators who are the experts in these areas, generating the data and make them accessible to the world. So with that, I'd like to introduce our speakers today. We have 2. Suna, Onengut-Gumuscu. I'm going to say your name wrong. I'm sorry, Suna.
That's fine. I did it fine. First one is okay.
Thank you. Who's going to talk about the background of type 1 diabetes and the overview of type 1 diabetes genetics. And then Kyle Gaulton's going to talk about functional genomic characterization of type 1 diabetes loci. And at the end, as time allows, I'll take you through the portal and our future plans. So with that, I'll hand the comm over to Suna, and she'll take it from here. Stop share. Oops, that's not on. There we go. Suna, are you available to share?
Okay, here we go. I'll put it on full screen.
Beautiful.
And everyone can hear me?
Yes.
Yep. Okay, perfect. So thanks for the great introduction into type 1 diabetes. So I'll just briefly go over type 1 diabetes and then the genetic studies that we've been carrying out throughout the years where we are, and then hand it over to Kyle for the functional part. And so type 1 diabetes is an autoimmune disease. It resolves with the genetics and environmental factors that play a role in an autoimmune response that results in destruction of the insulin secreting beta cells in the pancreas. And that leads to lifelong dependence on exogenous insulin. The highest rates of type 1 diabetes is observed in European ancestry subjects, and the lowest is in Asia, but the rates of type 1 diabetes is increasing worldwide to 2 to 5% each year. So, and we do see a seasonality of there's increased diagnosis in spring and autumn for type 1 diabetes. The onset is typically around 12 years of age, which is a little bit different than the other common diseases. So it's juvenile onset, but there are also type 1 diabetes cases diagnosed at a later stage in the 20s and in adults too. So as I said, environmental factors play a role in the disease development, and half of the other risk is genetic factors. And we see that genetic plays a role because there's aggregation of T1D cases in families, and monozygotic twins, the concordance rate is between 50 to 70, it's been reported to be around 50%. And again, one thing that's different from other common diseases or complex diseases is that half of the risk is in the HLA region for type 1 diabetes. HLA region has a pretty strong effect in disease risk. And we and others have shown that the HLA-DQ beta-1, DRB1, HLA-B, and HLA-A have independent risk variants. So if we take a step back and look at the progression of type 1 diabetes, here I'm plotting the time. And on the y-axis, it's the beta cell mass. So in genetically at-risk individuals with environmental triggers and autoimmune reactions initiated. And the biomarkers that we use and the hallmark that we use to identify the initiation of islet autoimmunity is are several autoantibodies against islet cells. And as the number of islet autoantibodies increases, a person's chance of getting type 1, progressing to type 1 diabetes increases. Usually if you have once 3 islet autoantibodies are detected, it's very likely that within a 5-year window, the individual will progress to type 1 diabetes. Around when 80% of the beta cells have been destroyed by the immune system, glucose metabolism is basically the body cannot deal with it and diagnose with type 1 diabetes. Now that there are studies detecting earlier progression of the autoimmune response, and hopefully we'll have a point where individuals 30% of type 1 diabetic cases, when they're diagnosed, they go into ketoacidosis coma. So the aim at this point is at least to try to diagnose type 1 diabetes before people are in the ER at an earlier stage. So if we look at the history of the genetics of type 1 diabetes, as I said, half of the risk, genetic risk, is in the HLA region, and in 1973, this was identified. And throughout the years, you can see in 10 years, approximately 10 years, then the insulin VNTR, variable number of tandem repeat in the promoter region, was identified to be associated with type 1 diabetes. Then candidate gene approaches identified several more genes. And so by 2006, we had 6 regions in the genome that had strong evidence of association with type 1 diabetes. With the advance in genotyping technology and availability of large cohorts and the cost of genotyping going substantially down, researchers were able to scan the genome on genotyping arrays and do a genome-wide association study. The first study that came out with type 1 diabetes data was the Wellcome Trust Case Control Consortium, and that was in 2007. Let me move ahead, okay. And that was 2,000 type 1 diabetes cases with 3,000 controls, along with other complex diseases was studied in this Wellcome Trust study. So, and in 2009, the Type 1 Diabetes Genetic Consortium, which was established to study the genetics of type 1 diabetes and had put a lot of efforts. Steve Rich is the primary investigator of T1DGC, so the director of T1DGC, and they put a lot of effort in collecting families around the world. So there were North America, Europe, UK, and Asia Pacific were the 4 networks for type 1 diabetes. And the initial aim was to collect parents and 2 affected SIPs with type 1 diabetes. And they later extended their efforts into collecting case and controls from lower incidence populations. and put a lot of effort into establishing collaborations with other cohorts that have type 1 diabetes cases and controls, and led to the GWAS publication and meta-analysis in 2009, where we identified 41 loci, and 18 of those loci were novel. So as Noel pointed out too, with GWAS studies now suddenly, in pre-2007, we only had 6 loci. And in 2009, so just in 2 years approximately, we now are up to 41 loci associated with type 1 diabetes. And with the GWAS, okay, great, we identify these loci, but we're basically putting a flag in the genome saying that there's something in this region, there's a risk variant. And what do we need to do? We need to go deeper into each of these regions and identify the causal variants or group of candidate SNPs, functional, the causal gene in the region, and the biology under it. Here, instead of putting chromosome names, as scientists, we tend to put the gene names here, but without functional studies to support it, these are the best guesses that we can do in the vicinity of the risk variant identified from GWAS studies. Sorry. So in the autoimmune community, we were not the only ones facing this challenge. Suddenly we have more than 40 loci, 50 loci to go in and genotype in a very dense array. So what we did is that we brought our forces together with other immune-mediated consortiums and created the Immunochip Consortium. It consists of 12 immunologically related human diseases. And we designed the Immunochip array that has 186 distinct loci. Here on the bottom, you can see these loci with number of SNPs in each loci that will map hopefully the variation, genetic variation and architecture in each region. The array consists of approximately 200,000 SNPs, and as I said before, in the GWAS and the genome-wide association array, you scan the genome, all of the genome, but it's sparse compared to the Immunochip when you go into a region. And now we suddenly have in 100,000 kb region, we have approximately 100,000 SNPs. So the aim of the fine mapping arrays are to capture the genetic architecture and hopefully help fine map and identify causal variants for the disease. So following the meta-analysis in 2015, we published our first Immunochip paper looking at 25,000 subjects. And in addition to previously reported loci, we've also identified 6 novel loci. And these data are being uploaded right now to the portal and will be shared with the community. And, following there's additional GWAS studies that have identified more type 1 diabetes risk. So we're now above 50, and actually I think more around 60 probably. And one thing with these genome-wide association studies, or the initial ones, is that they were heavily focused on European ancestry subjects, but we're trying to increase the diversity of, at least for type 1 diabetes, and all other diseases are doing the same in diversity of our study population. And the first largest, I guess, type 1 diabetes association study in a genome-wide scale was published in 2019. So what's coming up in 2021, let's jump there. And we have one under review manuscript for now that we've scaled up our study population. And as I inferred in my previous slide, that we're trying to increase diversity in our study in our type 1 diabetes research. So we've now genotyped more than 61,000 subjects, and have done a transancestral mapping. And here this is a slide just to show that, we do need to join our forces and bring a lot of cohorts for these studies. And I'd like to acknowledge all the studies that have contributed to the study, to this meta-analysis for type 1 diabetes. The samples have been collected in different parts of the world, and we have different ancestries in this new study. And we've now identified 75 loci. 36 of these have not been previously published at a genome-wide significance level. So we're pretty excited about the data and diving into each region and identifying the causal variants. And one thing that we've learned from these studies and previous studies too is that as your number increases, we have a chance of capturing independent risk variants in the same loci, basically. And this graph is a little bit showing that, where we have several, in 12 loci, we have 2 independent risk variants, and it goes down here in 3, 4, 5, and 8 independent risk variants. And functionally mapping these and understanding how they interplay in increasing or decreasing genetic risk is a pretty big challenge and will require a lot of functional data. I'll just skip to the next slide here. So what we've learned from the immunochip studies and fine mapping studies, and other groups have also shown this, is that for a lot of the common diseases, these risk variants do not lie in protein coding genes. They do not change amino acids. For type 1 diabetes, there's a few risk variants that do change amino acid, but out of the 75, it's only 3 or 4. Genes at this point, and most of the risk variants lie in the non-coding region of the genome. And for type 1 diabetes, when we overlaid the T1 decredible SNPs with annotations from the ENCODE data, we've identified that these SNPs are enriched in regions of the genome and immune system cells that are enhancers that have regulatory sequence. And what comes out from these studies, again, is that these are, courtesy of Stringrat and Andrew Wells, that a risk variant here can be regulating genes, way further down from where the risk variant is located. So future directions are obviously narrowing down the causal variants in each region, identifying these causal variants, in which tissue they function, in which genes they regulate, at which developmental stage it's important. And there's a lot of effort in now collecting functional data to tweeze and understand the biology underlying the autoimmune process in type 1 diabetes. There are now genetic risk scores out that are actively being used to identify at-risk individuals, and screening programs to look into high-risk type 1 diabetes subjects and if islet autoimmunity is initiated. Again, there's a lot of effort now for the genetics of type 1 diabetes, as I was referring to, that islet antibodies are the initiation of the immune response. So studies like TEDdy, TrialNet, DAISY, where they're following at-risk individuals and identifying these individuals as the islet autoimmunity progresses, is looking at, is it the same genetic factors that affect type 1 diabetes risk, or are there additional factors that play a role in initiation and progression of type 1 diabetes? And as I said at the beginning, I'm pretty excited about the functional genomic studies we're doing, and we'll at this point hand it over to Kyle to talk about those studies. And this is not giving, credit to everyone, but I try to at least list some of the leaders for the GWAS studies. And if there's any questions, I'm happy to take them.
You want to ask a question of Suna, just raise your hand and we can unmute you. I think that's how it works. If not, we will transition over to Kyle, and if you think about it as we're talking, we can circle back to it at the end. Thank you, Suna. That was excellent. It was fabulous, sort of setting the stage for the next, where we are with genetics and what's happening over the next couple of years.
Unmute here.
Yes, we can hear you, Kyle.
Okay, great. Can you see my screen?
Yes, we can.
Great. Okay, so I'll follow on from Suna's great presentation and talk a bit about efforts to then interpret genetic risk variants for type 1 diabetes using functional genomics. Talk a bit about the studies that we're doing, and then also touch on some of the same studies that Suna mentioned and what their groups are doing to follow up on some loci, as well as more general resources that are available to annotate risk variants. So, okay, so risk factors for type 1 diabetes, again, Suna touched on this, largely affect immune cell function, particularly genes that have been known for quite a long time in type 1 diabetes. Many of those affect immune cells, T cells in particular, but also and I guess probably thymus function and also antigen presenting cells. There's also likely a component that involves beta cells and intrinsic functions of beta cells themselves and participating in their own demise as part of the disease process, and several genes have been demonstrated to affect beta cell function in the context of type 1 risk, but then of course a majority of loci for type 1, as with other complex disease, the biological mechanisms, cell types, and the genes that are ultimately affected are largely unknown. You know, a large reason for this is that most of the loci are non-coding. There's a handful of loci that contain protein altering variants, so the HLA types, genes like PTPN22, TYK2, IFIH1, many genes that again have been known for quite a long time in type 1 diabetes, or in one particular example of UTR for the insulin BNTR. But beyond that, most loci are non-coding and likely affect gene regulation, and so interpreting these variants and how they function is quite a challenge. And so I've laid out here kind of general strategies that are used in the field to try to interpret non-coding risk variants for type 1 or other complex disease. So it's a large part of it, as Suna talked about, is moving from association signals to causal variants, and increasing resolution of fine mapping to pinpoint variants that are causal. Using that information to then define regulatory activity and the tissues and cell types that regulatory elements are active in, and how they intersect with variants that are causal for type 1, the effects of those variants on regulatory activity directly, then linking those elements over to genes that are regulated by these sites using a variety of assays, either 3D chromatin interaction assays, correlations between sites, EQTL mapping type approaches, and so forth. And then once you're able to link variants over to genes, then determining what the function of these genes is, what's their expression across different cell types and tissues, what's their molecular function in these cell types, and then developing more sophisticated models to understand their contribution to disease. So that's kind of the general framework that you know, we think about, and I think many groups use in terms of trying to interpret many of the risk signals for type 1 and other disease. So Suna mentioned, I included this exact plot in her presentation as well. So based on their studies from several years ago, there's a strong enrichment of risk variants for type 1 diabetes and immune regulatory elements, and probably the majority of loci that affect type 1 diabetes have some immune regulatory function. And so you're going beyond, you know, this finding, and similarly for other autoimmune disease that you know, many of those loci also affect immune cell function, trying to flesh out in more detail how variants affect immune cell function in different immune cell types. So I'll just touch on a couple studies that have been done by our groups as part of this consortium that we have. So one was the paper that Suna just mentioned from their groups, that's on bioRxiv, where in combination with their detailed transethnic fine mapping, they performed ATAC-seq in sorted CD4 T cell samples of both Europeans and African ancestry individuals, and then mapped QTLs for chromatin accessibility in T cells, and then identified examples of risk variants for type 1 diabetes that affected chromatin accessibility in T cells. So one example is that Very nice example, the BOC2 locus, where I think this locus is fine mapped down to a very small number of variants. One of the variants on the left has clear molecular effects on chromatin accessibility and T cells, where the other one doesn't, suggesting that this variant is causal or functional and likely causal for type 1 diabetes at this locus. It also is correlated with the expression of BOC2. Similarly, our group, another paper that's recently on bioRxiv, is related, but instead of sorting out certain cell populations and then profiling those in bulk, we use single cell assays to profile PBMCs. Generally, use that information to disentangle different cell types and subtypes, and then map chromatin QTLs within each of the cell types and subtypes. And so in doing that, we find thousands of QTLs for different immune cell subtypes, and then use that information to annotate risk loci for autoimmune disease, including type 1 diabetes. And I suppose great minds think alike, because we happen to had been interested in the same variant that was focused in Stephen and Suna's study at the BOC2 locus, where this variant, again, associated with type 1 diabetes, has a high probability of being causal for the disease, and is in a CD4 T cell site. It's fairly specific across other immune cells, and has molecular effects on that site, and further in reporter type assays affect the function of T cells. So two independent examples converging on the same variant, likely causal for type 1 diabetes and affecting T cell function. But then this is a bit different than some of the other diseases that we all study, and some of the tissues that we profile, and that there's a lot of resources that are available in the community, both studies people have done, but also like consortia type efforts to map regulatory information in immune cells. And so I'll just highlight a couple of studies that provide relevant information, studies that we've incorporated in our database and passed on to the type 1 diabetes knowledge portal. But there's quite a lot of information available, and trying to collect all this information and provide it to the type 1 diabetes portal is one of our big goals, but it's quite a big undertaking given that there's so much data. So one example on the left is a study that's in Nature Genetics, I think last year or 2 years ago, where they profiled 32 immune cell types that were sorted, and both in baseline and then also stimulated conditions. On the right, promoter capture high seed data linking regulatory elements to target genes in different blood immune cell types and subtypes. This is also part of the Blueprint Consortium. Other studies, so there's QTLs that have been derived in sorted cell types, and the T cells and monocytes for not just chromatin, but expression and histone modifications on the left, and then also on the right, eQTL studies of individual immune cell types derived from single cell RNA-seq. Again, these are just some examples of data sets that are available to annotate regulatory activity in immune cells, and there's quite a lot of resources available in terms of consortia and just large scale resources that contain a fair amount of immune cell data. So Blueprint is a large consortia that the specific focus of that was profiling immune cells. But then also resources that are more general contain a lot of immune cell data as well, such as ENCODE, Epigenome Roadmap, and the resources like Chip Atlas that reprocess data in Geo. Okay, so yeah, the goals are to make all these data available in the type 1 diabetes knowledge portal, in addition to the data that's being generated by our groups to annotate immune cell function. So then I'll talk a bit about beta cells, and like I said at the beginning, there's some evidence that variants affect beta cell function intrinsically and contribute to the onset of type 1 diabetes. A bit different to type 2 diabetes. There hasn't been so much evidence, although I'm not sure this is entirely the case, but certainly less so than for type 2 diabetes, where risk variants that affect beta cell function tend to not affect aspects of physiology like insulin secretion, things like this, but appear to be more related to how beta cells respond to environmental stressors that are presented to the cells in the context of development in type 1 diabetes. For example, pro-inflammatory cytokines. And so there was a nice paper that was in Nature Genetics, again, I think a couple of years ago, where they demonstrated by mapping regulatory programs in pancreatic islets in response to cytokines that risk variants for type 1 diabetes appeared to be enriched preferentially in sites that responded to cytokine exposure in islets relative to other sites in islets. And so we've also been doing similar studies in our group, and I know others in this consortium and other groups are working on very similar things as well. Basically, on the right, the way these studies work is that there's in vitro models of islets where you take primary islets and then you either culture them in untreated conditions, or you stimulate them in some way with culture with cytokines or other disease-relevant stressors. And on the bottom, if you have a risk variant, say for type 1 diabetes, that's highlighted here, the red asterisk, in normal conditions, the relevant site for that variant may not be off or may have limited activity. But in response to a stimulus like cytokines, the site becomes activated, and then the variant exerts its effects on that site. It's like a gene by environment interaction. And so by mapping the changes in these cells in response to these stimuli, we can get a better sense for how risk variants may be affecting beta cell function in T1D. So we have a study that's kind of building on what has been already shown in previous studies, that risk variants generally are enriched in sites that respond to cytokine treatment, and trying to go beyond that to defining the genes and relevant processes that are downstream of those signals. So this study's in preparation, but touches on multiple different types of data that I think are relevant to annotating risk variant function and understanding the mechanism of risk loci. So we take association statistics for type 1 diabetes, and we have maps of pancreatic islets in their epigenome, both baseline and cytokine stimulated for bulk ataxia, but then also increasingly single cell ataxia. We have measurements of variant effects on transcription factor binding in these sites using high throughput reporter assays. We link over from these sites to genes that they're potentially affecting using 3D interaction assays like high chip, high C type assays, and also correlations between pairs of sites via coaccessibility. We measure changes in gene expression, and then we also have data on when CRISPR screen of knocking out genes in beta cells and seeing which genes increase or decrease cell death in response to cytokine treatment when knocked out. And so here's just an example of layering all these pieces of information together to try to understand risk mechanisms in beta cells. So we have fine mapping data for T1D. Variants lie in sites that respond to cytokine exposure. Among these variants have evidence for affecting transcription factor binding. We link from these sites over to potential candidate genes using 3D interaction coaccessibility type information, which implicates several genes. This locus has actually been studied previously and argued that maybe this DEXY gene is responsible for the signal. But among the genes that are linked to the site is SOX1, which is a gene that in the CRISPR screen, when knocked out, increases cell death in response to cytokines. So we can layer all these pieces of information together to kind of hypothesize what we think the mechanism of this locus might be. May involve multiple genes, but it seems like potentially regulating SOX1 and its response to cytokine exposure and modulating cell death is a potential mechanism. And we've studied the SOX1 gene in more detail in beta cells and tried to validate that that's the case. And then more broadly, we find that so if you take genes that come out of this CRISPR screen and then you segregate those that when knocked out either increase or decrease cell death in response to cytokines, and then also further which genes change in expression in response to cytokines, we can find a subset of genes that preferentially localizes the type 1 diabetes loci, those that when knocked out, they increase cell death, and then also their expression is increased in cytokine exposure. So arguing that their function is protective generally against a cell death and cytokine exposure. And if we look at these genes here, many of those, the top ones, map to type 1 diabetes risk loci quite striking. So that gives you a sense for risk variants that may function in beta cells. And so finally, I'd just like to touch on very briefly, trying to identify additional cell types that may contribute to disease beyond immune cells and beta cells. And we've done a lot of profiling of single cell assays in relevant tissues and have derived these really nice maps of both immune and pancreatic cells, and tried to characterize the regulatory programs of each of these cell types. And we've intersected all these data with GWAS for type 1 diabetes. This paper is almost online. Actually, had submitted it to bioRxiv yesterday, but it's still being processed, but will be up relatively shortly. It's fairly large GWAS for type 1 diabetes, where we intersect it with all this regulatory information for various cell types. Of course, find very strong enrichment in T cells. And again, most of the risk of type 1 diabetes affects immune cell function, but we also find evidence at the bottom for additional cell types beyond T cells and beta cells. Something we thought was notable, where we found pretty strong enrichment for S and R ductal cells, which are exocrine cells in the pancreas. Some loci for type 1 diabetes are annotated specifically in exocrine cells that are not on in endocrine cells or T cells. And they're around genes that have very highly specific expression in exocrine cells, both S and R and ductal cells. Here's an example of this GP2 locus. GP2 is secreted with the zymogen granules from S and R cells as part of secreting digestive enzymes. And there's multiple examples of loci that are fairly specifically annotated in exocrine cells. So we think that may be a mechanism of how some of these loci work. As another example, CFTR, fairly famous gene, which causes cystic fibrosis, a risk variance for type 1 diabetes fall in a site that's specific to ductal cells, pancreatic ductal cells, relative to all other cell types that we profiled. This is a site that's linked to the CFTR gene in ductal cells. By CRISPR, we confirmed that element regulates CFTR and not other genes in the region. And that variant is a QTL4 CFTR expression in ductal cells. And kind of interestingly, is also associated with other pancreatic diseases like pancreatitis, pancreatic cysts, some evidence for maybe pancreatic cancer, all pancreatic disease of the exocrine pancreas. And so there may be a link between type 1 diabetes and these other pancreatic diseases via the exocrine pancreas, but that's just a hypothesis. Okay, so yeah, so that argues that there's a lot going on for type 1 diabetes, of course, as with other complex diseases. There's immune cells, beta cells, but other cells in the pancreas, exocrine cells, and so forth that are contributing likely to disease. And then there's also resources that are available, one of which I've highlighted here is PancDB, where we have been aggregating data from them to pass on to the type 1 diabetes knowledge portal, where they generate data in the pancreas from non-diabetic T1D as well as T2D individuals. They're led by Klaus, as well as Jason Moore at Penn as part of the HPAP consortium. And so allows you to profile changes in type 1 diabetes. And we have similar data ourselves in our group from the HERN consortium, so many consortiums, and from the NPOD repository, where we've also been profiling non-diabetic T1D, as well as autoantibody positive individuals who didn't have type 1 diabetes, but as Suna mentioned, many of these will have gone on to develop type 1 diabetes and understand how these regulatory programs change. So finally, passing all this information on to the type 1 diabetes knowledge portal is, of course, a major challenge. Those of you who are familiar with the type 2 diabetes efforts and the common metabolic disease portal know that a lot of the data that comes in terms of functionally annotating the genome comes from this resource that was developed in our group, the Diabetes Epigenome Atlas, where we have based this largely on software that was developed by the ENCODE consortium to store epigenomic, other functional genomic data. Originally it was for the type 2 diabetes AMP-T2D effort, but we've kind of expanded the focus now to more generally diabetes and its complications. And so there's multiple goals of this effort, but the primary goal is to provide, collect, annotate, and record this information, provide it to researchers, but really to pass it on to the knowledge portal, including the type 1 diabetes knowledge portal, in order to effectively annotate non-coding variants. there's a lot of data in the site that allows you to annotate regulatory elements, effects of variants on regulatory elements linked to genes, and understand the function of those genes. And so this data is passed on from our site, aggregated by Knowledge Portal, presented in various ways in the Knowledge Portal, some of which that I think Noelle and Maria will demo in a minute. I just have one example of how this information is used, and it can be used effectively to annotate risk variants. So this is the BOC2 locus I mentioned that both of our groups on this call have studied in some context, where if you go to the locus view of this gene, you search for BOC2 and you're interested in understanding the mechanism, shows you the association data, but then you can also load annotations that are aggregated from our Epigenome Atlas site. Filter those annotations, because there's quite a lot of them, by some evidence for enrichment for the trait, type 1 diabetes, and so that reduces it down to largely immune cell elements and T cell elements, and so you can overlay this with this annotation information for disease-enriched annotations and find that this variant, which I had mentioned previously, is in a T cell regulatory site, and so it gives you some indication of what this variant, this locus, but then also this variant might be doing functionally. And then on the variant page, if you look at that variant in a specific page, then you can see the annotations for that variant, and it's annotated in accessible chromatin and T cells. Okay, so summary. So of course, non-coding, risk variants for type 1 diabetes, primarily non-coding, many affect immune cells, but also affect pancreas. Similar challenges as all other complex disease of identifying causal genes and so forth. Substantial amount of data from T1D relevant tissues, particularly in immune cells, but also increasingly pancreas and the diabetes epigenome atlas that enables annotation of risk variants in the portal. So some specifics, it may be a bit different than some other traits, although I think it's probably similar to other traits as well, is that molecular effects at some loci, particularly beta cells, but probably also immune cells in terms of activation and response to autoantigens, are dependent on their interaction with environmental stimuli. So that means it needs to be considered in both genomic studies and how you effectively generate data that captures those effects, but then in cellular models to study the effects of these variants in genes to understand how they contribute to disease. Another challenge is that I didn't really describe too much, but most people probably know immune subtypes are very related and have highly related regulatory programs, which can complicate understanding true disease relevant cells, processes, and mechanisms, because there's a lot of sharing between the regulatory information. So disentangling which subtype of a T cell is relevant, it can be an import can be a bit of a challenge. There's also limited data on immune cells in the pancreas or pancreatic lymph nodes, especially during the disease process. There's some resources generating that, but most of the data that exists from immune cells is from peripheral blood. So how related is that to the cells that are actually contributing to disease in the pancreas directly is a bit of an open question. And then finally, as Suna mentioned at the beginning, substantial risk explained by MHC. Not many complex diseases, if any, that have risk locus with such a profound effect on as MHC on type 1 diabetes. And so these studies likely would benefit from being performed in specific haplotype backgrounds that allow you to capture those really large effects. Okay, thanks.
Excellent, Kyle. That was wonderful. Questions for Kyle before I take you on a little tour in our remaining moments?
I went a bit long, Noel, but
okay. It was worth it. All right, I'm going to take you on a little tour. I think, frankly, talks from Suna and Kyle I couldn't have asked for a better setup for sharing the portal now, because it's particularly as someone who doesn't study type 1 diabetes, I spend most of my time on type 2 historically. I was overwhelmed by the amount of rich data and different approaches that you all will be taking. And they're similar approaches, I should say, but different cell types and different communities. And I think we have a job to do, and I hope we can do it justice by representing these results in the portal in a way that is useful to the world. So with that, our goals for the type 1 diabetes knowledge portal are, sorry, I can't get my screen to share. Sorry, 1 second, guys. There we go. Can you see that? All right, excellent. So our goals for the type 1 diabetes are very straightforward, very similar to what we've had with the other portals, is to aggregate and harmonize genetic data that Suna was speaking of for type 1 diabetes and its related traits. Provide a resource to convene and represent the type 1 diabetes community. I think that's one thing I've learned from this is a whole set of investigators who have worked together in many places, but having a place for them to represent their authoritative data in a way that is sort of vetted by them and represented the way they want is really what we want to be doing with the type 1 diabetes knowledge portal. We want to implement and apply the best practice methods from the community and represent that on the portal. Integrate genomic data that Kyle spoke about and the experimental results from the functional studies, not only the ones that Kyle talked about, but the other ones that are funded by the NIDDK that I mentioned earlier. We want to bring them into this consortia and work with them to share their results on the portal. We provide representation and visualization of both the genetic and the genomic data, and also run methods that take into account both these data sources and represent the results of those on a portal. So with that, let me quickly show you the portal. So you can find this portal by two ways, simply by typing into your var, type1diabetesgenetics.org, and you'll come to the portal. Or you can type 1 CMDKP and come to the CMDKP, where you'll find the full resource of all the different disease portals. So I'm going to start there so you can see it through this lens. If you want to get to type 1 diabetes, you go to your disease-specific portal. Anyone can open and access this. You just need a Google account. That's one way by which we do this, or a Gmail account, excuse me. So here's your type 1 diabetes portal. Very similar to the other portals. But what's specific about this is when you're here, you're just specific for the data sets and traits and phenotypes specific for type 1 diabetes. If you want to see type 1 diabetes in the context of common metabolic disease, you can just go out there. And I'll go out there in a minute. So what we have specific here is custom content, information about the consortium here, information about how to collaborate, our policies, how we handle data, how we handle protection and privacy of data, how to contact us, all the sorts of things that you see in the regular portals. But let's talk a little bit about the data. So data that Suna mentioned, we're just in the beginning stages of type 1 diabetes portal. We just started. So for this, what we've loaded for you, particularly are renal, ocular, glycemic, and inflammatory bowel disease traits, because there's some interest in that from what they're speaking about the autoimmune component. But you can browse the different data sets that we have. You can browse them by data type or by phenotype. And for glycemic, you can see all the other phenotypes that are represented under that large category, that ontology we've created for you. So currently for type 1 diabetes only, there are only 2 data sets on the portal today available to you. One, Suna mentioned, the T1D-GC immunochip, and one, an unpublished data set from FinGen for type 1 diabetes. So for each of these, you can click on this and get full information about the provenance of the data, how it was done, any downloads if we have access to the full data, information about the cohorts, where you can view it in the portal, ancestry information, and so on and so forth. So that's sort of the genetic data. And what's going to happen in short order for that, and at the end of this month, we will have 18 new traits coupled with 7 new data sets available on the portal. And this will include traits including LADA, hematological traits. So this will be just also European and mixed ancestry, which would be great. Suna mentioned this data, and this is also data coming unpublished from Kyle's group for 500,000 samples on T1D. So a lot of this data will be available to the portal as of January 28th. So it's a little preview for you. So this will really increase the power of the T1D KP at that point. But let's talk about what we've got so far. So if you go to the portal, let's go to home, and you want to type in your favorite phenotype. You want to know what is known about, so for the genome-wide significant, so for the genome-wide evidence for a specific phenotype, you can type in type 1 diabetes, and you will get the Manhattan plot and QQ plot for the meta-analysis of all the data sets we have in the portal for type 1 diabetes as a phenotype, accounting for sample overlap. We call it our bottom line p-value. It's a way of distilling all the genetic data sets into one highlight finding. For this, you'll get the top variants associated with types of diabetes, the odds ratio, closest gene, consequence, specific pertinent information. You can filter it by any of these mechanisms at the top. You'll also get magma results, which help you get a sense of the common gene level associations for type 1 diabetes and any other trait in the portal. This is generated from summary level statistics. It's in exactly the way it does in the paper, but with summary level data. And many papers, the publication, they used individual level data. So you can see the gene level results listed here for you to peruse. You can click on that and get to the gene. I'll show you that in a moment. And what's nice about this is if you want to drill down to the data sets that go into the overall meta-analysis, you'll see here all the different data sets. As I mentioned earlier, you saw that in the data page. The 2 data sets that go into the meta-analysis are listed here. And then here, this is where DGA comes to the portal. This is where the annotations that Kyle spoke about the annotations that are going to be specific from the type 1 diabetes genetics community are going to be loaded into DGA and then represented here for you to peruse in concert with the genetic information. So they're listed here. So that's the sort of the high flag finding of a phenotype page. Let's talk about a variant page real quick. I'm cognizant of time, so I'll try not to go over. You go to a variant page, this is an example, a classic example on the type 1 diabetes portal. You'll get the variant, the closest gene, the predicted consequences from the variant effect predictor. You also get a PheWAS plot, courtesy of our Locus-Zoom colleagues, which will show you for every phenotype in the portal. This is again just for the type 1 diabetes portal. And what are the associations? You can also see this in a forest plot if you rather see it that way. And again, this is filterable and customizable. And for this, you'll know what other phenotypes are in the portal. And if you would like to view the data for that, you just go here, and that will drop down all the data sets that go into your overall meta-analysis for that trait in the portal. In addition to that, you will also have GF binding motifs. And again, the annotations will be viewable here as well for that variant. So let's go to the Common Metabolic Disease Knowledge Portal just to show you the last set of features. So if you want to see anything you know about type 1 diabetes in the context of all the other traits, this is where you want to go. You want to head to the Common Metabolic Disease Knowledge Portal. And I'm going to show the exact same gene that Kyle mentioned. It's kind of funny. It's like we're bakers, you know, he showed you how, and I'm going to show you what it looks like when it comes out of the oven. So this is VAC2, and what it does, if you type in the gene, it'll give you the region around it, and immediately shows for all the traits in the portal, which there are 325, what the top association. For this, the most significantly associated phenotype is hypothyroidism. But what I did already to show you this is I'm going to change this to just type 1 diabetes. Sorry, my dog is barking in the background, apologies. Anyways, change this to type 1 diabetes, the entire page filters for that. And you can also see the other associations in the region that are genome-wide significant. So in addition to type 1 diabetes, you have hypothyroidism, blood cell count, type 1 diabetes, BMI, FEV1 to FVC ratio, and so on and so forth. You can change the page to any of these if you want to look at the results for that. And again, they'll show you all the other phenotypes in the portal as well that are not significantly associated at genome-wide significance. So then you want to peruse the top associations for type 1 diabetes. They're here in a table that is again filterable and searchable. And what Kyle referenced, which is a new feature, which we're going to really spend a lot of investment in the next coming months, is our ability to visualize the genetic data with the genomic annotations in the context of credible sets, relevant annotations, tissues of interest, the best genetic data sets. And we call this the genomic region minor, or GEM, because hopefully it will be a gem of the portal. But it really is powered by Locus-Zoom, and the data comes from DGA and comes from our portal for the genetic data. And all the methods that are applied by our software platform that runs against the genetic and the genomic annotations and represents a visualization that you see here. Actually, I pre-canned this because it turns out it's the same exact thing. So to see what Kyle showed you in terms of overlaying the genetic data for the locus with the annotations, you can choose your annotation. There are a host of them. We're working on categorizing these because there's a lot you need to know what to look for. So you'll see some improvements to this over time. You can search by credible set, and you can also filter by enrichment. So things that are most significantly associated with your trait of interest. So what I did was I loaded exactly what Kyle suggested for VAC2, and you can see this is the same SNP he represented in his slides. And if you see here, you will see the CD4 positive T cell annotation right there that he mentioned in his talk. And so for this, you can load multiple annotations and see them in concert, see where things line up, see where the creditable sets are, and you can visualize a locus. And this is ideally a spot where we're going to start to allow you to prioritize genes for functional follow-up. So I realize it's 1 o'clock. That's a tour of the portal. Please take a look at it. Please send us feedback. What's not working? Please tell us if you want to collaborate, same data. What are we missing? What phenotypes would you like to add? Really, this only gets better because of the feedback we get from colleagues like yourselves. And any questions real quick in the interest of time? I realize that I'm over. Were there any questions? I see some chats. Suna, Kyle, absolutely fantastic. Excellent. That's great. Thank you, Phil. So actually, I will let everyone go if you are so inclined to take a look at the portal. Hope you can, one last thing actually, before you all break. We will have a release at the end of this month and some things to look forward to. right here, I will grab that, is several things across the platform. So across all of our portals, not only the CMDKP, we're going to have a lab resource. This is for novel experimental tools that have unpublished, fresh data, fresh methods applied to it that we're going to put in a labs page. We want your feedback. We want you to tell us what doesn't work, what you don't like. Ideally, these will eventually be integrated into the portal, but they're really a place for us to test out. So like Google had this for a while. In fact, Google Calendar was once Google, it was Google Labs. It was like new features. Do you like it? Do you don't? Use it, tell us what you think. We'll incorporate it if it's good. That'll come online at the end of this month. We'll have new methods, LD score regression and POPs run across all portal data, updates to the GEM module I mentioned, combined views of effector gene predictions for type 2 diabetes and several other traits. That'll be primo. Refine, refresh workflows for our aggregation tests, including TopMed and our 52K exomes, so you can have custom aggregation tests run. Association results viewed for complications in the context of type 2 diabetes, a way of viewing complications that we are experimenting with as we build the portal out for diabetes and its various forms. Predictions for CAD that we've been working with the Cardiogram Consortium on, that will be hosted with publication, and a brand new portal, a lung disease portal, where we will also have the COVID-19 host genetics data integrated and represented on that portal. So that's what's to come. Thank you for joining. I appreciate it. I'm sorry we went over time. Thank you to Suna and Kyle. That was wonderful. I appreciate it. And I hope to see you all next time. Take care.